All right. Good morning, everyone. We are going to be starting off the live stream here. I am speaking today presently at the Scripps Research Institute's Center for Metabolomics and Mass Spectrometry. And we have both people live here in our audience and online as well. There is, I don't know how many people we have online at the moment, but... Okay, I'm playing back from somewhere. Sorry about that. There's a bit of a delay. There we go. And we have three different speakers who are going to be talking for you today. First off is going to be myself. I'm going to be speaking about setting up the XCMS experiments and using XCMS online. And then we're going to go on to Xavier Domingo, who is originally from Barcelona, or Tarragona in Spain. He worked previously with one of our old postdocs from Scripps. And then we're going to move on to Erica at the final part of our talk, who's going to talk about XCMS systems biology. Erica has recently joined our lab and has made quite an impression as she's jumped on in. So without further ado, I'm going to jump into moving on to setting up our XCMS experiments. So setting up our XCMS experiments, what are we actually trying to do when we do XCMS? Well, hopefully what you've done is you've run off and you've done a lot of experiments, and you've sat down and you've thought about all of these experiments that you're going to do, and you now want to do some sort of comparative analysis. You want to be able to run some statistics, you want to be able to start to identify those different compounds, and hopefully you want to understand some of the biology, or at least what's going on in your system if it's not a regular biological setup. Now, one of the things I really like to do is I like to remind everyone what type of samples you're looking at. And so here's just a couple of simple TICs from a variety of different experiments that we have. And you'll see that you have many different samples here. All of these TICs are laid over the top of each other. And this is actually from a UPLC data set that we're seeing here. So this is ultra high pressure mass spec data, and it was run on a latest generation QTOF instrument, so it's very high resolution data as well. And you can see that our peaks are very nice and thin. We're getting somewhere between 2 seconds on the small side to about 30 seconds on the longer side retention time peaks here. And probably that first part of that chromatogram is very, very busy. There's a lot of information there. And that latter part of that chromatogram has less data in it. So we probably could have altered the chromatogram. This is one of the nice things I like about doing those TICs overlays, is that it very quickly tells you this type of information, and you can see what I'm going to expect from my experiment. So now that we're starting to remember more of what our data is looking like, let's have a quick overview and look at what XCMS actually looks like. We always start off with some sort of peak detection process where we go through our data and we find out what is a real peak and what is just noise. And then we have to do some sort of grouping of those peaks. So we take all of our peaks from all of our different samples, and we try to say that this is the same peak in a different sample. And we'll do that by trying to say that they're in the same mass and retention time area. And then once that's done, we have an option to do retention time alignment. I say an option because although we do it the vast majority of the time, it's not always necessary to actually do retention time alignment. Especially on some of those UPLC data sets, you'll notice that you get less than 5 seconds of deviation, and sometimes even less than a second deviation. And so in that case, the retention time alignment might actually just mess things up more than actually doing it in the first place. But the alignment will pull all of the those peaks back into alignment, and so we'll get better grouping, and we can honestly say that those peaks are the same. Finally, we'll go off and do some statistical analysis. Now, both myself and the other presenters are going to be talking very heavily about XCMS itself today. There are other platforms out there, whether it be vendor solutions or academic solutions, whether it be something like MS-Dial or NZMind2 or NZMatch. We're going to talk about XCMS and XCMS Online because, well, for myself, I'm a developer, and XCMS Online, of course, is developed here at the Scripps Research Institute.
The slides are shifting in the live stream, and so the left part of people's screens are cut off.
All right.
Just on the slides, the video looks good.
Just a moment. I'm just being told that on our live stream we have a little bit of a technical difficulty, so I'm just going to try and correct that and see if we can correct it. Apologies to the people who are in the audience at the moment. Just give me a quick moment. All right, so we will do this. Very sorry, everyone, for this short technical difficulty. If you just give us a moment or two, we're just correcting a issue with the way that the PowerPoint screen was being pulled up. Hopefully this is better now. Let's just have a quick look. I'm just going to pull ourselves back in here. So, all right, perfect. Due that this is doing this edit live, I'm going to leave it as such. I hope everyone's all right for that. All right. So, as I say, we have different types of software out there. And in XCMS, we actually have a variety of different job types that we can go up and start using. And we do this to try and make things quick and easy and get you moving on the path to running your experiment as soon as you possibly can. And we split these up into fairly logical settings. So the first is the single class job, which, just as the name sounds, it's where you have either a single file or you have a group of files which don't have any class to them. And you want to run this and get some sort of peak list, get some sort of feature list back from this so that you can start to see what's happening in your data set. Then there's the pairwise analysis jobs. Now the pairwise analysis jobs, what they do is we're now starting to compare two different classes between each other. So we have an A set and a B set, and we want to see is a particular feature different in one class versus another class. So this is your normal wild type versus knockout type scenario. Then we have multi-group, multi-class jobs. This is where you have a much more complex setup. You have many, many different more classes. Something like a time course, or something where you're sampling from multiple different sites or wells, multiple different tissues maybe. Something much more complicated than a simple A versus B type comparison. And these work really, really well, and I think this is where the multivariate stats really start to come into their own. Then there is the Meta XCMS and multimodal. Both of these are starting to get a revised look at in the latest bit of our work. And This is the Meta XCMS. What we've now starting to change this into is being able to go back and look at old samples, old jobs, and be able to run that data set against a newer data set. And we'll have future streams about this and talk much more in detail in the coming months. The multimodal jobs allows you to be able to combine multiple different jobs, so a pairwise or a multimodal group, and look at two different modes that you've done it in. So maybe you've collected in both positive and negative mode, helix negative and reverse phase positive, and you want to try and get the biggest overlap in your systems biology. I don't want to talk too much about the systems biology because both Xavier will touch on it very briefly, and Erica will talk about it in much greater detail later today. So what does XCMS Online actually look like when we start to get into it? There's this very nice interface that you're brought up with. Everything is online. You can use the website xcmsonline.scripps.edu, which you probably know about if you're watching this stream or here in the audience. And as you go through, of course, I've logged in here, as you can see. I go through and I say I'm going to create a job. As soon as I've created a job, I select the type of job that I want to create. I'm going to say a pairwise for this. I go ahead and I'm brought up with, I don't know why that didn't work. So once I say I'm going to create a job, I have a selection that I have to make, and then I can say that I want to load a new data set. When I load a new data set, I'm given, my windows are out of order, and I apologize for that. A new window pops up where I can load in my samples, and I can make sure that all of my samples are loaded up. XCMS Online will take any raw data file format from Agilent, Bruker, Waters, Thermo, and ABSciX, and of course any open vendor file format, any open data format such as MZML, which is of course the newest, MZXML, MZData, and the much older NetCDF format. If you are running GC data, the only way to be able to process data is converting to NetCDF data. Just a caveat on that. So once we have our data loaded in, we'll have something like this view where we're told that the number of files that we have, the data set name, and we'll get a data set ID. If we're happy with that, we can go on and we can start to say that we've done that for both our first data set and our second data set. And then finally, we're going to run with our different selected parameters. And we have a list of different parameters that we can choose from. We're going to go and select a particular, just the first one, so the HPLC QTOF data set. And we can start to go through our different parameters and have a look at what those parameters look like. There's a View Edit button, and a screen like this will pop up where we can look at all of those different parameters. And for each and every single parameter that we have a list of what those different options are. And a little bit of a reminder. Now, there's a couple of things I want to make note of when you are choosing your parameters. You want to make sure that you've chosen the statistic type that you want for that particular job. The statistic type is not saved with a parameter, it's saved with the actual job. So if I rerun that parameter set again, the statistic type will just drop back to whatever the default is, and I probably won't get what I was expecting to have. So I want to make sure that I'm going to have the chosen statistic type that I want. Now the other thing as well, just to notice or remember, is that if I go and I pull this back up, as some people have said to me to check that the system took that, it will reload back up the default, and you will lose your saved parameter or saved statistic type that you just put in. So just know that it has taken that statistic type and don't try and check it, because you'll just go into a constant loop. The other thing that you want to do, and we added this, I guess about maybe 2 years ago now, is the different biosource type. And you want to make sure that the biosource that you're going to use is either the organism that you're looking at, or at least the very closest organism that you can get to. We have about 7,500 different species in there. Of course, there are many, many more that we could possibly put in, and we're always trying to make this number bigger. So if we're all happy with all of our data sets we've uploaded, all of our parameters that we've chosen, we've got our right statistic type and our right biosource, then we can go ahead and we can actually submit our job. The system's going to check quickly to make sure that we have the type of data that, or that everything is the way we look. We can go and change our job name here quickly as well if we want, and then finally hit submit. Once we hit submit, it's going to send it off to our XCMS inline grid here at the Scripps Research Institute, and it's going to start processing this data for us. It'll send you an email when it's finally finished and say, hey Paul, your data's finished, and you can have a look at it. And you can see previous jobs that we have here as well. And while this one's running, we're going to take a quick look into what it actually does. Now, like I said at the very beginning, this is what XCMS does. It does some peak detection, it does some grouping of features, retention time alignment, and then the statistical analysis. These are all of the different parameters that you've just put in that set there. And so let's take a quick look at what some of those algorithms are actually doing behind the scenes there. So as you go through, one of the first things to know is that peak detection is actually quite a difficult thing for the computer to do. For us, of course, it's quite easy. We look at something, we know that it's a peak, I can tell you it's a peak. It's a little bit harder for us to start to integrate that actual peak. But for the computer, it has to find a way to decide to do that. So one of the things that the Stefan Newman's group did back in 2007, I believe it was start to say that peaks can look a little bit like missiles. Missiles fire up into the air, and then they come back down and they hit a target. As they go up into the air, they move from side to side, they avoid objects and satellites and planes, and then, as I say, they come back down and they hit the target. Well, an LCMS trace is much like that. And so it elutes off the column, it comes up, it wiggles from side to side, it gets a very accurate mass over charge, and then it comes back down into baseline. And that's exactly the same thing that we're seeing here. So we like to detect things backwards because it's a little bit easier doing it that way. There's a slower slope on that side rather than a very high initial kickoff. And so that's exactly what we do. Once we're able to find where something goes from baseline up and then comes back down, we can find the beginning and the end of that. And now what's really neat about this is that we have started to find places where we've been able to dynamically bin this. So here in this image, what we're seeing is we're seeing a little bit of a different different way of looking at the data. And so we have time on the bottom, so that's retention time in seconds on your x-axis. And then on your y-axis, instead of intensity, what we've put there is mass over charge. And so you can see here, and I'll do this both on the board and on the screen for everyone, you can see that there are various different compounds coming out here. So you can see that there's one compound here with a line of different peaks which are being found, or centroids. And then here's another set. And there's another one which is very, very close here. I'll just do that on the board, this one right here. And again, what we're able to do is we're able to identify that that is indeed some sort of unique feature, and that also this is another one right here. And what happens is we actually track from point to point to point, and the system has to say it has to be a detectable and traceable path from one to another. And I can't deviate in mass too much from one side or the other. And so as I do that, you can see here that it's going to start to wiggle through this, and that's exactly what it does. And what's very nice here is because I've done this backwards, I was able to find the second one as well. So after that's done, I've done this algorithm, I've done this pass over the entire file. I do this file by file by file, each independently. And once I've done this, I found places where I think that there's probably a peak. And I found many of these places. And as you can tell, this is an original figure from the original Trotenham paper. And I can tell you that there is a feature here, 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 but I don't know any of their integrated intensities. What I can do now is I can put some sort of filter. Stefan and his group decided to use wavelets for this, which fit very nicely. And I can put a wavelet over the top of these areas and find out their intensity. This is very nice because I've dynamically binned over my data set. I haven't had to go point by point by point by point, like many algorithms still do even today. So then once we have all of our different peaks, we know where these peaks are in our data sets, each file individually. We want to start to say, I know that this peak is the same as another peak in different files. And so I'm going to go off and I'm going to start doing this. And I'm going to start to pull these different, I'm going to do an overlay and check that if they're very close to each other in both retention time again and master charge. So again, retention time and master charge here. Master charge and retention time, that this is what I'm going to term a well-behaved peak group, and this is a very particular term in XCMS Online, or in XCMS. And what these well-behaved peak groups mean is they are groups that I have one peak from each file inside the system. And I do have an option for missing peaks and extra peaks in case I need those. And this is used during the alignment system. So let's take an example here. Let's say I have 10 peaks, or 10 samples. If I have those 10 samples, for a well-behaved peak group, within that mass retention time box, I expect to see 10 peaks inside that box. If I have a missing of 1, I can have 9. If I have an extra of 1, I can have 11 in there. And so missing an extra, missing of course happens because one sample for whatever reason didn't detect that peak. An extra is maybe another peak from a neighboring feature group moved over into there. Once I've found all of these well-behaved peak groups, I can then use the median for each and every single one of those different groups that I've found, and I can start to do some sort of regression where I can start to align sample by sample to each other. And in XCMS, this is done via a local regression function. Now, when you're finished with that algorithm, you're going to come up with something that looks a little bit like this. And here you'll notice that there's a lot of circles and squares and triangles and pluses and a bunch of other different signals. And each one of those represents a different well-behaved peak group, a different anchor in your regression algorithm that you've used. And what's really nice and important to see here is that you have a good spread of peaks across, or of those anchors, across that entire retention time area. They're not just grouped on one side, and they're not grouped on the other side. If they were grouped on the beginning and at the end, then of course what you would really have is a linear alignment between those 2 points, and that's not what you want. The more groups you have, the better your spread is going to be. Now, this may not be the case. You may have chosen to do the other algorithm, which is Orbiwarp, which I'm not going to go into detail here today, but we will have future streaming events and presentations to talk about that. Now, as I said, it takes the median retention time of each of those well-behaved groups, and it does it between each file. and you want to make sure that you have a good spread of those. Now that we've found all of our peaks, we found that those peaks are the same between our samples, we've aligned the ones which have drifted, we may come into the instance where we have exactly what we really want in a versus B type comparison. And so when that happens, we have this beautiful peak here showing that there is some compound which we probably are quite interested in. And in our other sample class that we're comparing to, there's absolutely no intensity. And so in many softwares, what you would get is you would get a simple NA value or a 0 value or something. In XCMS, what we do is we go back and we fill in the baseline. And so that way you get some sort of number compared to your actual full result. This really helps your statistics. It really helps your overall analysis as well. So now that the job's actually done and it's gone through, it's done all of that processing, this is the type of view that you're going to get. And I'll just take you through very quickly. So here you have your normal TIC, you have your retention time alignment, your retention time deviation plot, your corrected retention time plot, and you can ask yourself if these two look very different than each other. Then you have some of your cloud plots. Again, I'm not going to go into detail about this. For those of you who are streaming online, there's much more information about that online. For those of you here, feel free to ask me about that at the end of today. The MDS plot and the PCA, these are both multivariate methods to look at the data. And then down the side we have our results table, our cloud plot, our heat map, our PCA, and our connection plot. So if we go and we look at our results table, you'll see that there is many different features that we have here, and we can go through and just arrow through each one of those, which is very, very nice. And then for each one of those will be shown our EIC, our spectral plots, and then a Metlin search on what that compound could be. And this is a very nice way to kind of quickly get through your data and decide what's happening. Then we have our cloud plot, and again, I'm not going to go into too much detail about that to try and move on to some of the other slides. But very briefly, it is a TIC which is mirrored, so that we have retention time on the x-axis, and then we have mass to charge on the y-axis, so we have from 0 to whatever our maximum was, and then 0 down to whatever our maximum was. And you can see that there are green and red circles. Those green and red circles are feature groups which are particularly highly differentiated between one set or other set. They're either upregulated on the top or downregulated on the bottom. And the different sizes are how significantly different it is, and then the color is the fold change on that. And in this view, you can play around with the different bits. You can click on the different bubbles, and you'll get told not only what its exact mass to charge and retention time is, and p-value and fold change, but you can also see a EIC and spectral plots off to the side of that, which is very nice. And as I said, that's the cloud plot. And then, of course, we have our interactive PCA, where you can change some of the different scaling metrics that you're going to use, and the heat map, which is a nice kind of quick overview of your data. And then finally, there is the systems biology workflow, where we use the Mummy-Chog algorithm, which was published first by Professor Lee out of Emory University. and that was that paper, I believe, was quite some time ago now. And this is very nice. I'm not going to go into any detail about it because Erica will speak much more detail about this later today. So before I end, do we have any questions? And also, please remember that there's a wealth of more information on the XCMS Online Institute and on our FAQ site. Do we have any questions from our audience here? Erica, do we have any questions from online?
We had one. I had Dr. Domingo help me answer it, but
okay.
We'll say it verbally as well. We were asked, is the range in binning dependent on the type of instrument?
Range in binning. So
when you choose your binning sets for parameters, this came up with the peak detection.
Sure. Right. So when we're detecting the that trace, what happens is it is instrument dependent, and so the more it is not the we quote it in ppm. So when you have a trace and the system tells you that your vendor tells you that you have 5 ppm accuracy. That's 5 ppm accuracy on the top of your peak, not scan to scan to scan. And what the algorithm is actually doing is it's tracing from one scan to the next scan. And so it's asking, if you imagine it's like a horse with blinders on, and so it's saying, can I see 5 ppm this way? Can I see 5 ppm this way? And if your scan-to-scan mass-to-charge ratio is more than 5 ppm, and it happens to be outside of that, then it will not be able to detect that trace. You will not detect that peak. So it's better to set it ever so slightly wider than it is narrower. If you set it too wide, then the algorithm will have to make a decision whether it goes left or whether it goes right. But I'd rather it have to make that decision than trying to just not find your trace in the first place. The other thing to remember as well is this is not scan-to-scan variability at the top of your peak. This is scan-to-scan variability at the bottom of your peak as well.
You have a few more questions coming in.
Okay, sure.
Which sample was set up as control sample in the cloud plot as default? So one can know conclusion about up and down regulation. So how do you assign which one
first, is the... I believe it says on the, as you're setting it up, but the first sample, I believe, is the control, and the second sample is your, experimental.
Is it possible to edit or define the color of each single chromatogram in the TIC results?
Of each one, off the top of my head, I don't know. I don't believe it is. I know you can change the color of the circles that you get.
Can we do optimization of LCMS parameters like retention time correction, mass error improvement during online settings? Can we do an optimization?
You can. So with XCMS Online, because we have, I think we have 28,000 odd users at the moment. Our system's always turning over, and because of this, we have to put some restrictions. So we have to put I think we currently have a 50 gigabyte limit, 50 or 25 gigabyte limit, I forget which it is now. And then we also restrict users to one XCMS job at a time, and you have to wait until that XCMS job is finished. If you're happy to do that, then yes, you can do a optimization and run through your different parameters yourself. There has been talk to put that on our list for it to automatically go through that. Unfortunately, our development list is always very, very long. All right, in the interest of time, I'm going to pass over to Dr. Domingo, who's going to be speaking to you about annotation and systems biology. Xavier?
Thank you, Paul. Good morning, I'm Xavier, and I'll be talking about annotation. So Paul has talked about how to convert the raw mass spectrometry data into and use XCMS Online to convert these raw mass spectrometry data into a list of features. And we're going to use this feature list to find dysregulated peaks, dysregulated features among conditions. And then we're going to use, find or from the peaks corresponding to those dysregulated features to identify what are the dysregulated metabolites underlying our samples. Now, it's important that before we do any identification of our peaks, we notate that data. And it's important for two reasons. The first one is that we need to know what feature is what. We need to know that feature is a protonated species, that feature is an adduct, is an insource fragment. And second, we need to know if we want to have a putative idea, putative identification of the identity of that feature. So we can enable a systems biology or analysis at the systems level of our samples, of our biological question. Now, XCMS Online covers or allows annotation of metabolites, data annotation at different levels. Some are already implemented, some are under development. The first level is feature annotation. I'm going to explain all these levels throughout my presentation, just giving a brief overview. The first one, as I said, is feature annotation. This is enabled through the package camera that is already integrated in our system. We're developing another package called Everest for advanced metabolite annotation. We have already implemented an insource fragment based strategy to identify or at least retrieve a more robust putative identification of the underlying metabolites in our samples. We are working on the use of retention time for the same purpose, to rank at least the putative identifications that we find or that we have for each feature. And finally, we're also working on a cognitive analysis of our samples to try to leverage the information from scientific literature to, again, annotate better or to get a better idea of what we're dealing with in our samples. All these levels combined allow us to get a more robust putative idea of what are the observed dysregulated metabolites in our samples, so we can combine that information also with other omic strategy, other omic analysis, and enable in turn a systems level analysis. Let's take one step back. Let's go back to LCMS data. LCMS data is highly dimensional and redundant. What does this mean? Let's put an example. Let's say that we have one metabolite, one metabolite with our mass spectrometer, one single molecule, but we're going to see different peaks that stem actually from the same metabolite. We're going to see the protonated species, we're going to see some isotopes, some adducts, and some common losses, and even insource fragments. And so, although we're using ESI, electrospray ionization, which is a soft ionization technique, we're going to see some fragmentation. And this is actually an existing phenomenon. In fact, common losses are as a result of, or are produced, are observed as a result of insource fragmentation. They're not fragments per se, but they occur as a result of insource fragmentation. So, as I say, for one metabolite, we have different peaks. So we have a redundancy, as I call, and we have, so of course we're observing thousands of metabolites in our samples, so you do the math. We're going to see we have a highly dimensional data set, a highly dimensional list of features in our sample. So it's something to that we want to or we need to take into account. This is a very simple example. This is using scheme. I want to show you two real examples of two metabolites in real samples. This is an example of two metabolites in, as I say, in real samples, and you see that we observe lots of peaks, lots of features. Now, some of them noise, of course, but some of them are not. Some of them are features that stem from the same metabolite. So we're observing different features that all belong to one single molecule. So there is a redundancy. And this is one of the main objectives of data annotation. One of the main objectives is try to group all the features that stem from the same metabolite. And why do we need that? Why do we need algorithms that effectively perform that process? Because we still try to answer how many compounds are we profiling in a typical untargeted metabolomics experiment. We're still talking of features when we do an untargeted experiment. And at this point, we should be talking about compounds. And this is one of the problems why we cannot answer this question, because there is a lack of good or at least effective algorithms capable of grouping all the features that stem from the same metabolite and telling us how many metabolites are we observing in a typical metabolomics experiment. Again, metabolite annotation aims precisely at that, trying to group all the features that belong to the same metabolite and pick up the right feature to fragment the peak corresponding to that feature and compare the fragmentation pattern with our database or with spectral reference databases. We want to pick up the right features because we will, if we have multiple features for the same metabolite, we want to choose the feature. We wouldn't want to choose an insource fragment. We don't want to choose a loss. We want, if possible, choose the protonetic species or the protonetic species that is going to give the most relevant structural information of that molecule with mass spectrometry. Now, there are different strategies for metabolite annotation. We covered or we explained these different strategies in our last review. This year, Erica's gonna send you a link in the chat of the review. And there are different, as I said, I'm gonna cover these 5 strategies. There are some other strategies. For example, now there's an emerging the CC as the correlation cross-sectional values that we can also use to annotate molecules, but I'm gonna focus today just on those ones. The first is MS1 pseudo-spectral extraction, the use of atom mass rules, the use of biochemical knowledge, leveraging insert fragmentation, and finally use retention time calibration to annotate data. Let's go one per one. Let's first start with MS1 pseudo-spectral extraction. As I said, we have different features that stem from the same metabolite, so we need to group, we need an algorithm that is capable of distinguishing what features stem from each metabolite and extract what we call an MS1 pseudospectra that is composed of all the features that stem from the same and each metabolite. There are different techniques to perform that, mainly 2. The first one is to use the peak shape, to correlate the peak shape of the different features. If 2 features show a high correlation, show a strong correlation, we assume that those peaks or those features stem from the same metabolite. If they show a weak correlation, we assume that those 2 features do not stem from the same metabolite. This strategy was integrated within the package camera. It's an R package, and it's also integrated in our XCMS online system, and it starts calculating or computing all the correlations among all the features and constructs a network, and then it extracts what are the features that are correlated and therefore assumed to stem from the same metabolite. Now, this has one problem, that is that if 2 metabolites, as I'm showing here, if the features of 2 metabolites are coeluting, then they are going to show a strong correlation, and the algorithm is going to think that they stem from the same metabolite, where in fact they do not. So this is one of the most important limitations of that approach. There is another approach, which is called peak abundance correlation, which works this way. Let's imagine that we have one sample, and we have 4 peaks, 4 features, A, B, C, and D. A and C belong to the same metabolite, B and D belong to another metabolite. And we're going to have these features here in the sample, but then in another sample we're going to have the same features with different intensities, and again for as many samples as we have. The features, the intensities they're going to vary, but the intensities between the ratio between the intensities among features that stem from the same metabolite is going to be the same. So in other words, if we compare the area of A versus C and the area of B versus D across the samples, we're going to observe a very high linear correlation, a strong correlation. And we can take this fact in our advantage to determine what features stem from the same metabolite. If we compare features that do not belong to the same metabolite, let's say B or A and B, we're going to see a poor correlation, and therefore that means that they do not belong to the same metabolite. So once we know what features stem from the same metabolite, then we can start computing mass distances between the features and compare those distances to known mass from adducts, losses, and so on. And we can use that information to annotate the features and say, well, this feature is probably a protected species, this feature is an adduct, those are the isotopes, and common losses. We cannot annotate with this strategy insert fragments because they are specific. There are fragments from the molecule, so therefore they are specific or more specific, let's say, for each molecule. Therefore, we cannot apply those rules for metabolite annotation in those cases. Now, again, there are a lot of features that are going to remain unannotated because there are complex relationships occurring, complex phenomena happening in our mass spectrometer. And this is something that they studied in this interesting article by Majumder and colleagues, where they propose, well, if we use more complex relationships, we can maybe get a better idea of what these other features are. Now, they also explain or discuss that the more relationships that you take into account, the more chances you have to find false positives. Of course, the more the likelihood is that you find relations between two unrelated molecules that correspond to, any other mass or rule that you add or you consider. There is another annotation strategy now based on biochemical knowledge, which was introduced by Xu Li. Paul was talking about it a moment ago. Eric is going to go through it again in his talk. It was introduced by Xu Li, and we adapted also this algorithm, and we integrated that algorithm in our XCMS online platform. And people now recognize the value of this work because it kills two birds from one shot. What it does is it gives you a putative idea of the metabolites that you have in our samples, and it also projects those metabolites into pathways, so we can have an idea of what's going on in our system at a systems level, right? So, and it's based on one assumption, which is the following: if a list of putative identifications are true, this should reflect a biological activity, and thus show an enrichment on local pathway regions. What does this mean? Let's show this with an example. Okay, let's say that we have a feature list with 10,000 features, and we start identifying just by taking into account the accurate mass, and we identify all these features putatively, and we have a list of putative metabolite identities. Let's go one by one through all the dysregulated features that we have. Let's assume that this feature here could be any of those metabolites, and let's assume that it could be this 4-methylamino butyrate. Okay, let's take this compound and project this compound into its corresponding pathway. Okay, we're gonna see that this in red is our dysregulated features, and there are other compounds close to this. Exactly. This compound, the red is our dysregulated feature that is projected into this pathway, and there are other molecules close to this molecule, but there is no feature that could be attributed to any of those metabolites. There is no dysregulated feature that's important, a dysregulated feature that could be attributed to any of those metabolites just by accurate mass search. Okay, let's assume does not make a lot of biochemical sense, right? So let's assume now that this molecule or this feature could be valine. Okay, we take valine and we project valine into its corresponding pathway. Now we see that there are other dysregulated features that could be attributed to other metabolites that are close to this valine, to this compound. Therefore, this makes more biochemical sense, or makes more sense from the biochemical point of view. So we can assume that these features in fact valine, and that the other features, other dysregulated features, are those features that they claim to be. So this is how this algorithm works. It putatively identifies the features, and at the same time it gives us an idea at a systems level of what's going on, right, in our samples. So another strategy is leveraging insert fragmentation. We're thinking how to annotate those insert fragments. As I said before, we cannot annotate insert fragments with typical annotation rules because they're unique from each metabolite, right? And we were thinking how to do that. And then, okay, we said, okay, we have melon. We have the largest collision dissociation spectral database, mass spectral database. And we have, for all the molecules that we have in the database, we have data from 0, 10, 20, and 40 volts. That means that we have low collision energy spectra in our database. And we know that those insert fragments that we observe in typical MS1 experiments are the same fragments that we observe in low energy MS data. So in other words, if we compare the MS spectra of valine, of low energy spectra, with the MS1 features that we observe in our XCMS online experiments, there's going to be a match, and we can use that to identify, or at least get a more robust idea of what are we observing, what are the dysregulated metabolites that we are observing. And we were also thinking, or we ask ourselves, how often this insert fragmentation occurs. And to answer that question, we processed all melon. At that time, we had we processed all the molecules, all the low energy spectra that we had in melon at the time. This is also something that we reported for our review that Erica sent you the link a moment ago. And we came up with this picture. And here, this picture shows the in the y-axis the percentage of metabolites that have 0, 1, 2, 3, 4, and so on number of fragments in both positive and negative mode. And you can see from here that only less than 10% of the molecules in melons, 10% of the molecules that we usually observe in our samples, have non-insert fragments, whereas the vast majority, more than 90% of the molecules, have at least one or more or multiple insert fragments. So that means that insert fragmentation is actually a real phenomenon. It occurs, and we want to take this phenomena as an advantage instead of a drawback by, as I said, comparing these insert fragments with melon to have a more robust idea or a more robust putative identification of our molecules or features. Let me show you two examples of that. Let me show you some examples of two compounds two features that we set of features corresponding to two different compounds. We retrieve those features with XCMS Online, and then we use melon to find the identities of those features by, again, using in-source fragmentation. And as you can see, the fragments match, relative intensities do not necessarily have to match, but we can, again, use in-source fragmentation to find the different molecules that we have in our samples. Now there is another strategy based on rotation time calibration. Again, we were collecting thousands of spectra with Melin. At the same time, we are also collecting rotation time information. So we were thinking how we can use that data, how we can use all this information that we have available to do something useful for metabolomics, especially for untargeted experiments. And well, we have rotation time calibration information, but of course the problem is that we cannot, those rotation times are unique for a specific chromatographic method. So how can we make this scalable? How can we make this rotation time information universal for other users? And well, interestingly, some studies, some researchers have shown that even if you have the data from one specific chromatographic method, you can use that data and project that data into another chromatographic method. And this has been shown here in this study by Stransop and colleagues, where let's say that, let's focus on this panel here, in panel A. Let's say that we have two chromatographic methods. In one chromatographic method, we have 50 molecules. In another chromatographic method, we have another 50 molecules. And they share, let's say, 25 molecules. So we can use that information to construct a curve, to construct a function, right? And then we can project the data from the other 25 molecules here and the other way around. So in other words, we can have rotation time information from molecules that we have not identified from another chromatographic method to ours, and the other way around. And they even built a database to do that. So if you have information of a rotation time in your chromatographic method, let's say that you run an untargeted experiment, but you identify some molecules, then you can upload this information into the database. This database is going to find what's the chromatographic method, the most similar chromatographic method to yours in the database, and then it's going to give information of the rotation time of other molecules that you have not identified, so that now you can, or at least get a more robust idea of what those other features that you have not identified are, thanks to the use of rotation time. So in conclusion, we're working towards advanced metabolite annotation methods and strategies. Again, XCMS Online now has allows or enables feature annotation by the use of the R package camera, and we're working on another strategy for that purpose. We have also developed an in-source fragmentation algorithm to use in-source fragmentation to get a more robust idea of what are the molecules that we're observing. We're working on the use of retention time to also run or rule out the different candidates for different features that we observe dysregulated. And finally, Eric is also working on a cognitive analysis algorithm to try to use or leverage information from scientific literature to annotate, again, the features that we observe in our samples. At the end, we want to combine different levels, or by combining different levels of annotation, we're able to get a robust idea, or at least, yeah, as I say, a more robust idea of what are the molecules that we are observing, so we can combine that information with other omics strategies and get a mechanistic insight of our biological samples or our phenotype. And that's it. If you have any questions, I'll be happy to answer.
We have 3 questions so far. One is for both you and Paul Chahi. They asked, at the end of Paul's talk, could you explain more about the retention time correction plot? How do you define that retention time is well aligned from the plot?
Right. So just to jump back on that quickly, from the plot of that retention time deviation, what you're looking for is that you have plenty of well-behaved peak groups, anchors across that retention time alignment, and that retention time deviation, so down that y-axis, is an acceptable range of what you're wanting. So that is going to be dependent upon whether you're looking at UPLC or HPLC. You'll notice that the points will exist further outside what the line is. The line is where it is pulled back to post the alignment. And so you're hoping that all of your lines are under the acceptable deviation that you have. So to speed things on, I'm going to now move any questions further to Xavier. I know that there were a couple.
Okay, Chavi.
Mhm.
About in-source fragmentation,
Mhm.
I identified an annotated peak relevant in the PCA as being actually the base peak of a compound, only because I knew about the possibility of this compound occurrence. Is there a way to determine this with confidence using Metlin instead? Do you-
I'm not sure if I completely understand. So, to identify the nature of that, okay,
Yeah,
this
so
person know that this is a peak.
yeah, so could they annotate their significantly dysregulated
Sure.
peak
Mhm.
in Metlin?
By using the insert fragment, you can annotate, because that's the correct term to use in that situation, so you can have an idea of what this molecule could be. But if you really want to identify this molecule, what you have to do is, of course, fragment the specific peak corresponding to that feature by using tandem MS, and then try to find, again, using Melin, but now using different, not just low energy, but the most best collision energy that you can use for that specific peak, and identify that spectra now by using tandem MS information, proper tandem MS fragmentation.
Okay, I think that starts to get at this next one.
Mhm.
It says, and for matrix generation, for multivariate analyses, how this in-source fragmentation peaks can be treated. They can be considered in the same peak list of all other regular peaks.
Exactly, that's a very important question. I mean, when you do multivariate analysis, one of the things that you want to take into account, or one of the problems, is that there is a high dimensionality. Now, by using in-source fragmentation information, or this in-source fragmentation information that we provide, you can maybe think of removing those in-source fragments and just take into account the proteinated species, or maybe you want to average or do some sort of transformation in your data to try to remove, or at least have a less dimensional, or have less features, so you can work better with your data when you use multivariate analysis.
And Paul just typed an answer into the chat, but if you're not reading the chat, the final question is, great talk to you both, enjoying it so far. Do you think that in the near future, Metlin could include fragments from lipid compounds?
Absolutely. Melin already includes fragments from lipids, and actually, lipids have a lot of insert fragmentation. They have a lot of insert fragments, so you can use that information also to not just try to get a better idea of what these lipids could be, but also as maybe with respect to the other question, try to remove all these features that are going to be redundant for the same metabolite. So, yeah, the answer is yes. Thank you very much. Yeah, I'll pass it to Erica.
So I'm Erica Majumder. As they said, I'm the newest postdoc here at the Scripps Center for Mass Spec and Metabolomics. And I'm excited to share with you stories about using our systems biology platform. So XCMS Online has this systems biology portal that has been very useful to me. My science mostly relies on microbiology in complex environmental systems, and I've really found this to be a useful portal, so I wanted to share with you those experiences today. I'll be giving an example of how we have aligned metabolomics data with transcriptomics data to get at real mechanistic understanding of our particular condition. If you need more detailed procedures of our experiments today, there are 2 papers out of our lab last year, one in Nature Methods that describes the systems biology portal in general, and then another one in Nature Protocols that will walk you step by step through how to use the platform in greater detail than I will demonstrate today, but also feel free to ask any questions. So, systems biology is a field that is looking at biological phenomenon on a larger scale level, and it's combining information from multiple skills across biology. And that usually involves the use of computation or bioinformatics as well. So some people take a look at molecular networks and build them up to cellular networks, and then finally organ or tissue networks. If that's not really your thing, then some people look more on these other levels by combining DNA and RNA and proteins and getting up to the phenotype level or the cellular level to understand what's going on in their system. Traditionally, people have been using more genomics or proteomics-based techniques, but our lab has been pioneering the incorporation of metabolomics into systems biology workflows, where we use metabolites to identify the key pathways that are connected to the phenotype that you are observing in your system. And this can be any type of biological system that you're interested in, from bacteria to humans and everything in between. So, metabolomics-driven systems biology is how our systems biology portal works. That means we identify the dysregulated metabolic features from a pairwise XCMS job, meaning those are the ones that are significantly different in your condition. So those are the metabolic pathways that should be tied to them. Paul went over in detail how this part of the system works, so I will not repeat it, but we get those dysregulated pathways from our dysregulated metabolites from projecting them, as Xavier told us about. And then we also have the ability to overlay and integrate other forms of multi-omic data to create a true snapshot of what's going on. Each different omic technique brings a different level of information to the question, and so by combining them, we can have a much larger sense of what's going on. Additionally, as with any metabolomics, identifying the compounds you're using are important, but Xavier and Paul have both gone over how we do that. So I will be giving you both a tutorial and an example of the work that I've been doing and how I personally use the systems biology portal. The experiment I'm demonstrating today is an experiment where we took a field-isolated bacterium. The nasty-looking ponds you're seeing are actually the waste retention ponds at Oak Ridge National Laboratory in the 1980s. And this is where all the leftover waste from enriching uranium during the Manhattan Project was dumped. And that has unfortunately started to seep into the groundwater. So you have quite high contamination there of uranium and technetium and lots of other stuff. But of more significant note, actually, is that it's the site with the highest nitrate contamination that's been reported globally. So there's a great deal of nitrate contamination. That's not only a problem for nuclear waste sites, But as we've started to apply more fertilizer to agriculture and other things, we have a very significant increase of nitrates in our groundwater throughout the country. So we really want to be thinking about how do we remediate that groundwater, how do we understand it. However, these environmental systems are incredibly complex, and there's many multivariate factors, and so we've started to use omic technologies like metabolomics and transcriptomics to understand the flux of these contaminants and how the microbial communities that naturally exist there are responding to these contaminants, and how we might actually be able to use them to remediate them or to move that. And so therefore metabolomics is helping us understand their metabolism. So we isolated a bacteria from these highly contaminated areas and have been growing it and studying its metabolism under conditions reminiscent of the field site so that we can understand how to best stimulate remediation here. This particular bacteria is a nitrate-reducing bacteria, which is great. It's helping us remove the nitrate contamination, and it's highly tolerant of the metals. However, the problem is that the nitrate-reducing bacteria are inhibited in their growth, and therefore the cleanup of the nitrate by another predominant group of bacteria at the site, the sulfate-reducing bacteria, and they're inhibited by these reduced sulfur compounds that the sulfate reducers give off. And so we've been looking particularly at the mechanism of the growth inhibition of the nitrate reducers from those reduced sulfur species across several time points. So metabolomics is giving us a sense of what was happening, and the transcriptomics is confirming those pathways and looking at it on the gene level. Before I get into the experimental details, I also wanted to provide some considerations that I've learned over time about multi-lab planning. So this experiment that we did, the samples were harvested in Oak Ridge, Tennessee. I was working in my lab at Missouri at the time, and we had to send it up to our collaborators in Seattle, and then to Berkeley, and then down here to San Diego. So as you're doing these multi-omic experiments, there's a lot that I've learned to consider. You need to think in advance about which omics are most appropriate to answer your experimental question. And I see my slides have gotten a little jumbled, and I apologize. I think that was a PC to Mac conversion. All the samples need to be done on the exact same time with the exact same starting material and with sufficient replicates. And it's very important to make sure you have enough material for all the replicates for all of your multi-omic samples at the same time. All of these omic technologies are at such high resolution that even small differences in the preparation can prevent you from getting a biological understanding out of your results. Similarly, you need to think about the fact that DNA, RNA, peptides, and small molecules all need different treatments and preservation techniques. You cannot do the same thing to all of them. And you need to have a consistent way to normalize your samples across the techniques, or you won't be able to have a quantitative or even a semi-quantitative view of how much something was changing, which is how we dissect information out of multi-omic or systems biology type investigations. And finally, is just a very simple thing, creating a naming chart and having a sample tracker can save you a lot of headaches when you're coordinating across many labs and people and states. Okay, Paul covered this in great detail, so I will just refer to that you need to run a pairwise job in XCMS systems biology or in XCMS online. Then the key thing for systems biology is to make sure that once you're setting up your job that you modify your parameter settings to select your biosource. Paul did a great job of telling us about the biosource, but we want to make sure that we truly select that, because the biosource is what holds the genome of the organism, so the genes and the proteins that are present, as well as a particular selected group of metabolites. So if you were studying a mouse or a bacteria and you left it on the human pathway, your data would be incomprehensible and you're not getting the value of it. So this is a very key parameter to make sure that you select your biosource. Once your job has finished processing to get to your systems biology results, this is what your landing page looks like, and you want to click on the systems biology green button. That will bring you to something that looks like this. I will highlight later what the pathway cloud plot and the predictive metabolite results look like, and I will just say briefly that all of the pathways and metabolites are text searchable, and you can search in the text box if you're looking for something in particular. When we zoom in on that central part, this is telling you your top dysregulated pathways, and it's sorted by p-value. You can also see which genes and proteins are found on the pathway. All of the metabolites that are on the pathway is the all metabolites, and then those metabolites which were found in your data set are the overlapping metabolites, and how significant we think those are comes into the p-value equation. When you hover over the pathway, that has a direct link to biopsych, so if you click on that, it will take you to biopsych, and you can learn about what that pathway is. And similarly, if you click on the link of the overlapping metabolites, it will show you what all of those metabolites are. And the total metabolites on the pathway are also visible and linked. So you can navigate to many different parts of the systems biology portal at one time. In my system, this is the screenshot of exactly what I get when I open mine up. These are one of the tests we did that were cysteine inhibition of our nitrate-reducing bacteria, and we're trying to find out how the cysteine is inhibiting the growth. So we're looking at which pathways have been shut down by the introduction of the reduced sulfur contaminant. And of interest is the top one, actually, which is this isoleucine biosynthesis pathway. So when we saw that, that started to give us a hypothesis, because it's been reported in other systems that cysteine toxicity is linked to problems in branch-chain amino acid biosynthesis. So that was giving us our first hypothesis. The rest of them make a lot of sense to us. In general, we're seeing pathways that are linked to central energy metabolism and just the general growth of the cell, and we know from the phenotype that the cells are not growing well. So it's confirmation that we're seeing different types of anaerobic metabolism and central carbon metabolism knocked down in this situation. Similarly, we're treating with cysteine, and so it makes sense that our other sulfur-containing amino acid, methionine, is giving us problems, and other sulfur-related pathways like coenzyme A biosynthesis. Then the next feature that you might want to check out in the systems biology portal is the pathway cloud plot, which is accessible by clicking on the other green button. And this is the general page of what you will get when you open your pathway cloud plot. It is sorted by p-value. So we have on the bottom is the percent metabolite overlap. So how many metabolites were found compared to the total number of metabolites available on that pathway. And on the y-axis is the negative log of the p-value, so how significant that is. So in general, we are interested in finding those metabolites, or the significantly dysregulated pathways are up on the top right corner. Are the ones that we're looking for. Again, everything in here is hyperlinked, so you can always find out more information about your data. If you click on the pathway or hover over it, it'll give you a zoom in and tell you more information about it. You can also live toggle your data and adjust it by p-value. And similarly, you get a full list again of those results tables telling you about which pathways are significantly dysregulated. So here is my data again from our field isolated nitrate-reducing bacterium. When I have it, the p-value set at a decent significance of less than 0.05, I still have a whole ton of pathways. And it's not telling me a lot, particularly since many of them are clustering together. So I reduced that filter to less than 0.01. So now I'm really looking at my very significantly dysregulated pathways, and these ones with higher overlap are the ones I'm going to start to try to focus on. So what's jumping out at me again is that isoleucine biosynthesis. I went and looked in at this cardiolipin biosynthesis, and it's actually not that interesting. It's a very short pathway, and so it's only pulling one metabolite. So I'm not going to focus on that. And the ones down here, again, are telling me that the cells just aren't growing very happy, which we observed from our known phenotype. The next thing you can investigate in the systems biology portal in general is which of those metabolites are actually found on your pathway. So when you click on the overlapping metabolites, this is the page that it brings you to, and there's a lot of information tucked into these pages. So there's a pie chart, again, just representing how well that overlap looks. You get the list of metabolites on the left side here. I'm trying to point with the cursor. I apologize if you can see it. And then we have, and when you click on the Metlin ID or the KEGG ID that are found next to them, you will get the Metlin information about that metabolite, and similarly what KEGG has to tell you with what's going on there. This little green box over here is the feature number that XCMS Online assigns to you, and when you click on that, you get linked back to the chromatographic and the mass spec data for that particular feature. And there's other things in here that I will discuss shortly, but it tells you about the up and down regulation of each individual feature and the fold change and the p-value for each feature, so you can start to evaluate in general how the entire pathway was responding. Again, so this is my actual data now, not a general example, and so I've pulled out that first pathway that I'm really thinking might be the significant one, this isoleucine biosynthesis. Again, you get this tiny table with so much detailed, packed information in it. So let's take a closer look. These are my metabolites that I'm seeing. What I like is that it's quite consistent in that the dysregulation for all of them is up. So that's telling me that this whole pathway was up. And to refer to one of the questions that was asked earlier, sometimes I don't pay a great deal of attention to which order I put them in, and so it's hard to know which one your control and which one your example are, but you can always look in the results table and find out which sample means up. So in this case, the way I set it up was in the regular, or in the non-treated condition. So this pathway has been shut down in the contaminated condition, and looking at the fold change in a quite significant manner. Those are pretty large p-values, particularly later in the pathway. So again, this is supporting my hypothesis that the branched chain amino acid pathways are downregulated and are partially responsible for the observed growth inhibition under cysteine-treated conditions. One of the last things you can look at in the systems biology is the predictive metabolites, and these, instead of listing by pathway, this is the inverse. It lists all of the metabolites that were used that strongly pulled the different pathways. These are the ones that gave us the indication of the pathways that we're seeing. The way and so it'll tell you the name of the metabolite and then all the names of the pathways that it found, and again is linked to the feature information and the mass spec data. So no matter where you are in the systems biology portal, you can navigate between these and always look back at your raw data and evaluate the features that you're looking at. In my field example, how I sometimes use this portal personally myself is when I'm in the predictive metabolites one, I'm going to sort it by fold change to see the most significantly dysregulated metabolites that are indicating my particular pathways. And in this case, this was my top dysregulated predictive metabolite, and it's involved in a lot of DNA and RNA and nucleotide processing. But since it was pulling so many pathways, when I saw those top dysregulated pathways, that actually tells me that a lot of them were because of this one metabolite. And so I can sort of eliminate those and say that, yes, like DNA synthesis was definitely downregulated, but it's not the biggest thing that's going on, which gives further credo, again, to our hypothesis about branched-chain amino acid biosynthesis. So we're using all these different clues to start to put it together. And all of that information that I just told you about was only from the metabolomics data. Now we'll talk about how I've integrated transcriptomics data and use that to really address our hypothesis about branched-chain amino acid biosynthesis inhibiting, being the responsible pathway for the growth inhibition in our nitrate-reducing bacteria. So in our XCMS systems biology portal, you can integrate either transcriptomics or proteomics data. Transcriptomics data looks at the number of gene copies that were produced at a particular time point, and analogously, proteomics looks at the number of protein copies that are available at a time point. One caveat on our systems biology portal is that this is not a quantitative integration at the moment, but only a overlay of just pathway matching. So that gene is supposed to be on that pathway. If you see it on your data, we match that up. We're hoping to have that be a more quantitative integration in the future. So how do you do this? One key piece of information that isn't currently well laid out on our website is how to format your multi-omics data, so either your transcriptomics or your proteomics data, before you upload it to our portal. You are responsible for filtering your multi-omic data, so your proteomics or your transcriptomics data, to only have your significantly dysregulated genes or proteins. And so you have to decide what that means to you. This is my actual data, or actually I should say my collaborators from this institute, from systems biology, they did the transcriptomics for me. This is their data and how we started to process to only our significant genes from our sample. But this is really a very important thing to do. Secondly, you need to convert the data naming or the nomenclature of the genes or the proteins to match the systems biology portal. We use the biopsych nomenclature for mammalian systems. That usually means the gene ontology. For bacterial systems, it mostly follows the locus ID, but you have to look at which version of the genome our systems biology portal has and you're using. In the case of the most closely aligning genome to our field isolate, there had been a newer version of the genome than XCMS systems biology had, so I had to convert the nomenclature within the locus IDs. So just be aware of that. And then finally, you want to reduce your gene or your protein list to a one single column CSV with no heading for the most easy upload. For the multi-omics one, under the green systems biology buttons, there's a little plus sign. You click that to upload your multi-omics data. And you simply do that by clicking upload list and then indicating which type of data that is. And then you want to click the run matching subjob, and that's what will overlay our proteomics and our genomics data onto our metabolic pathways. So when you do this, this is a general example. Now the overlapping genes and overlapping proteins, if you had both genomics and proteomics, are populated, and you can view those results in the same way that we did for the overlapping metabolites. And again, are linked to biopsych, so you can always find out more information about them. In my data, this is what we found out. you can sort those overlapping genes to find out which pathways had the best overlap between our metabolomics and our transcriptomics. So when I did this, I found that the transcriptomics was agreeing with a lot of our energy-related pathways and a lot of our sulfur-related pathways, and again starting to hint at our amino acid biosynthesis hypotheses. I apologize, this is really small. The table looks good on your screen, but not as good when you copy it into data. But overall, we can actually see that there's decent overlap between the transcriptomics pulled pathways and our metabolite pulled pathways. So that's giving us new overlap, but also new insight into what's happening mechanistically inside of the cell. This is a very busy one. This is just to say that not in our XCMS Systems Biology portal, but as a way of validating our findings, my collaborators at the Institute for Systems Biology in BioPsych itself took the transcriptomics data to pull pathways instead of what we do where we take the metabolomics data to pull pathways, and then they overlaid my metabolomics results onto those transcriptomics pulled pathways. And again, the primarily thing that we were founding was this branch chain amino acid biosynthesis pathway, as well as good agreement on downregulation in anaerobic respiration. So that was very nice confirmation that using many different types of multi-omic platforms and sorting your tools can give you new pieces of information but point you in the right direction. So the outcome of that particular experiment, or as of right now, we actually still have a lot more ongoing studies with that, that we see good agreement between our metabolomics and our transcriptomics. The two different omics are giving us new insights into it. We generated this hypothesis that amino acid biosynthesis was one of the major impacted areas that was causal to the growth inhibition. What's very cool is that I don't have the data to show you yet because it's hot off the presses, but we went back into the lab and we tested this, and we actually saw that branch chain amino acid supplementation was able to rescue that growth inhibition phenotype. So the metabolomics data coupled with our transcriptomics was like dead on pinpointing us to one of the mechanistic reasons for our growth inhibition in our field isolate, which is quite cool. And we're connecting that pathway observation to the mechanism, which is pretty neat. And so metabolomics-based systems biology was able to connect phenotype with underlying mechanism. I've given you an example of a relatively obscure nitrate-reducing bacterium from the soil, but it actually has a lot of important implications. But the same can be done for your data in any type of biological system you have a question about. So to summarize our webinar today, I totally forgot to mention that this overlay of the metabolomics and the transcriptomics was actually only possible because of the annotation platforms that Xavier talked about. Based on those matching, we're able to look at global profiling of these systems. So everything was really coming together today, both in terms of the XCMS Systems Biology and our new powers with automated annotation are helping us to view our large-scale systems biology data in a much more robust manner to get at mechanistic understanding. So taking that back to summarize today's findings, we saw a lot of fun things that we're able to do within XCMS and Metlin. Paul introduced us to the platform and really how to get going with your XCMS jobs. Xavier enlightened us on how all of our different new types of metabolite annotations and these pipelines that are making metabolomics approachable for all of us. Thank you, Xavier. I'm not a computational person, so I greatly appreciate those. And I hope you found our applications within systems biology interesting and really starting to see how metabolomics has become a robust technology that lets us pinpoint mechanistic understanding. And that's all. we're happy to address any questions from the three of us, and we hope we didn't go over too much.
So, Erica, you do have one or two questions. One question from Laura. She is wondering, let me just put it back up to where it was. Sorry, not from Laura. Let me just find the question again. It is if I have classes within the same data set, season, and a plant part, which job would you choose to do XCMS? So she has multiple different classes within a data set. This is from a environmental response with the same species seeding.
Okay, sounds like a really cool experiment first. Yeah, when you have large data sets like that, the type of job you're going to use will depend on the question. Will depend on the question that you're asking. So, if you want to know what's different between them, then you're going to use our pairwise jobs, and you'll have to do multiple versions of pairwise jobs. In the example I was showing today, I actually have about 10 different pairwise jobs that relate to that, but I was only showing you one of them. So that's a way to individually dissect them. If you want to look more at a group, you can use our multi or our meta jobs, and multi-group ones will tell you what's different between those ones, and the meta jobs will tell you more about what's similar between those jobs.
Right, and that was a much better answer than I had written down, so thank you for that.
I'm sure it's not.
And in addition to that as well, briefly in your own words, because again I wrote something, since the software has so many different metabolites that it has to choose from, how does it find out which is the correct one for the pathway?
That's a great question. So I'll give a short explanation, and then I'll let the guys chime in if they find a more computational need. So whenever you're making a metabolite identification or annotation assignment, and in metabolomics I've learned recently, there are standards and levels for the identifications, and you can use different pieces of information to help you sort out the answer. In the systems biology portal, we should stress that the metabolite lists on the pathways are putative identifications, and that you need to go back in and do formal identification processes on the metabolites that you think are very important to your study. However, so what MummyChalk does, or how our systems biology algorithm assigns the putative identification, is it actually uses the pathway data that's predicted from the genome of the organism as a piece of orthogonal data in addition to the mass and other mass spectrometry based data to give that putative assignment. But you do need to do formal identification on that feature to confirm it. But when we see many metabolites behaving similarly that should be assigned to the same pathway, that gives us a lot of confidence that that's probably the correct answer, and that's where that associated p-value comes from.
And one last question is, have you used other softwares for integrating multi-omics data, and what were them?
So,
what were they?
So other than pulling into Biopsych, I personally have not used other integrative softwares yet, but I've been making a list of them because there's actually a great deal number of them, and they're emerging, and some are better for either field. In the chat box to the webinar in a few minutes, I can post a link to some of the other ones or to a couple of review papers I know about that describe some of the different other multi-omic platforms that are out there.
Okay. Right, and on that note then, I am going to say thank you very much to everyone who is both here at Scripps with us and who are also online. Again, we will be, this is a little bit of a pilot as well as the post-streaming event that we did previously. And we're hoping that everyone likes this, and with some of the feedback from last time and today, we will hope to have this as a bit of a series at some interval to be decided upon shortly. So again, thank you very much for everyone who is online and here. We hope to see you shortly. Cheers.
Cheers.